Hello students, I am Dinesh Udupi. In this video, we are going to continue with the second part of immunology topic, and we'll be studying the types of immune responses, the two types of immune responses, and we will be covering only the syllabus which is prescribed by our college. Now, as we already seen, this is the overall syllabus which we will be covering in series of videos. Now let us start with the study of lymphoid organs. Now, as we already know, like different type of organ systems, there is one system called immune system in our body. And this immune system is mainly made up of different types of lymphoid organs. Now, lymphoid organs are mainly associated with the production of cells which are involved in the immunity, and also they are associated with the fighting with the pathogen. So the production of cells and how these cells function, so they are taken care by these lymphoid organs. Now we identified two kinds of lymphoid organs. One is primary lymphoid organs, which is represented by thymus and bone marrow in human beings, and the peripheral lymphoid organs like lymph nodes, spleen, gut associated lymphoid tissue, and mucus associated lymphoid tissue, etc. Pairs patches are also there. Now, what is the difference between primary lymphoid organ and secondary lymphoid organ? Now, primary lymphoid organs are the ones where the primary lymphoid organs are the ones which are involved in the production of blood cells like WBCs. So the production of cells and the maturation of cells associated with immune system occurs in the primary lymphoid organs, whereas peripheral lymphoid organs are associated with the functional aspect of these cells. So actually the cells which are produced in the primary organs, they move to these peripheral organs and they wait for the antigens or pathogens to come in to their contact. Now let us see some of the organs here. The lymphoid organs, you can see the tonsils and adenoids which are found nearby the salivary glands, and lymph nodes. We'll see lymph nodes. Now lymph nodes are the nodal structures which are found in the lymphatic system. Now lymphatic vessels and lymph nodes are part of the lymphatic system. Thymus, now this is one of the important primary lymphoid organ, and then the bone marrow. So here we have just shown one bone. You know, there are so many bones, large sized bones which are found in your arm, in your leg, etc. So almost all the large sized bones, they have marrow inside them. So that marrow part of the bone is the lymphoid organ. Then we have a spleen in our abdominal cavity, and Pairs patches here in the alimentary canal, and there is gut associated lymphatic system associated with the gut. Then appendix, even though they consider it as a vestigial organ, primarily maybe during the early course of evolution it was mainly associated with immune response. And all over the body we find lymph nodes, and lymph nodes we mainly find in this region around the salivary glands, in the armpits, and in the groin region. Maybe you know that whenever we get infected, sometimes we develop swellings in this part of the body. So either in the armpits or in the groin region, we may find the swelling that is a kind of immune response which has resulted in the inflammation of these lymph nodes. Okay, now this is Pairs patches. I'm sorry, this is thymus histology showing cortex, medulla, etc. Let us not worry about that in this video. And the bursa of Fabricius. So here I have mentioned bursa of Fabricius as one of the primary lymphoid organ. Now what is this? This is the organ which is found only in the birds, like hen. And you can see here, you can find this bursa of Fabricius just above the cloacal region. This is not found in mammals. In mammals, it is replaced with bone marrow. Now, bone marrow is a very, very important lymphoid organ because it is the place where the blood cells are produced. Of course, it is associated with the production of both red blood cells and white blood cells. And white blood cells are the ones which are mainly associated with immunity. Here you can see they produce hematopoietic cells, which are considered as the stem cells, giving rise to all other types of blood cells. And this one line will give rise to cells like, it's called myeloid progenitor cell. It will give rise to platelets, eosinophils, basophils, neutrophils, and monocytes. And also the most important type of cell, red blood cell, which is associated with transportation of oxygen and many other functions. And another line is going to produce the cells associated with immune response. And the stem cell or the progenitor is called lymphoid progenitor cell, and it will give rise to two important types of cell. One is T cell and another one is B cell. So these B cells are associated with humoral mediated immunity, whereas T cells are primarily involved with cell mediated immunity. Of course, they interact with each other in both type of immune response. Now let us move on to the next slide. And here it is just a picture showing the lymphatic system. We are more familiar with the circulatory system involving heart, blood vessels and blood, arteries, veins, etc. Now we have another system called lymphatic system, which is made up of lymphoid organs, lymph vessels and lymph nodes. Now lymph basically refers to the fluid which is found in the tissue. So this is the fluid which is almost similar to the blood, but it remains outside the blood vessel. It is found in the tissues. Now this also plays a very important role in many physiological aspects. But when it comes to the immunity, the lymphatic system, lymphoid organs along with lymph vessels and lymph nodes, they play a very important role in the immune response. Now we can watch a small video about this lymphatic system and we'll come back.
Did you know your body has its own version of a sewer system for removing unwanted toxins, waste and excess water from the body? This is called our lymphatic system. Like our blood circulation, the lymphatic system is made up of millions of little vessels that branch all around our body. But whereas arteries and veins carry blood, the lymphatic vessels are much, much finer and carry a colourless fluid called lymph. Our lymphatic system has three main duties. 1. It contains white blood cells called lymphocytes, which are used to fight infection. 2. It acts as a one-way drainage system, transporting fluid from body tissues into the blood circulation. 3. It gets rid of waste products produced by cells. As blood circulates the body, plasma leaves the blood vessels and travels into the body tissues, delivering food, oxygen, and hormones to the cells. This plasma becomes tissue fluid and surrounds all our body's tissues. It then collects waste products, excess water, and toxins from the cells. 90% of this fluid passes back into the blood circulation. However, 10% of the fluid is left behind and is known as lymph. We therefore need a system to drain this lymph fluid, so that we don't end up as a swollen fluid-filled balloon, hence the lymphatic system. The lymph fluid drains into lymph vessels, where it then travels to lymph nodes, which are found all around your body. To stop backflow, the lymph vessels have one-way valves and muscular walls that contract to force the lymph forward. This is happening in millions of little lymphatic vessels in your body every minute of every day. The lymph nodes filter the lymph, destroying or trapping anything harmful. The lymph nodes contain white blood cells called lymphocytes, which attack and break down bacteria, viruses, damaged cells, or cancer cells. This filtering and destroying is an important part of our immune system and stops nasty substances being fed back into our bloodstream, which would make us sick. The waste products and destroyed bacteria are then carried in the lymph fluid through larger lymph vessels up to your neck, where the thoracic duct empties the lymph back into the blood circulation. The bloodstream then removes the waste products from the body as it does with other waste. As well as the lymph fluid, our lymphatic system also includes a few organs: the spleen, thymus, tonsils, and adenoids. They all have roles in helping our body fight off infections. Unfortunately, like any sewer system, things can go horribly wrong. Sometimes we get infections that cause the pumping to break down. This can cause your arms or legs to swell to huge proportions. Sometimes the debris or other bad substances can make it past lymph nodes. Sometimes cancer cells do this and spread the cancer to other parts of the body, which obviously isn't good. For now,
Okay, it is just a list of primary lymphoid organs and secondary lymphoid organs. I already told you in humans the primary lymphoid organs are represented by thymus and bone marrow. Okay, these two. And so I already mentioned that these are the organs where the cells are actually developed. And secondary lymphoid organs include spleen, lymph nodes, tonsils, appendix, Peyer's patches, etc. And these are the sites where the cells which are produced by primary lymphoid organs, they enter into these secondary lymphoid organs and they wait there for the antigens or the pathogens to enter into the system. Okay, so this is a small overview of different types of cells of immune system. Now, this is multipotent hematopoietic stem cell which is going to give rise to all kinds of blood cells. Now remember, some of the cells will retain that potential and they become stem cells. So that they can become a permanent source of the cells, blood cells. Now this stem cell will give rise to two types of source cells. One become myeloid stem cell and another one will become lymphoid stem cell. So we already discussed it just few minutes ago. The myeloid stem cell will give rise to different type of cells. So the end product is platelets, red blood cells, erythrocytes, basophil, neutrophil, eosinophil and monocytes. Now these are the pathways through which they follow their end product. Now, the cells which are mainly associated with immune system are these macrophages. So these macrophages are capable of engulfing the pathogens like bacteria and other things. So they will directly through the phagocytosis, they will destroy the pathogens. And these are the exclusive cells like natural killer cells, T lymphocytes, B lymphocytes. The same B lymphocytes will be converted into plasma cells. And remember, natural killer cells are also the cells which will destroy the pathogens by the process of phagocytosis. Okay, let us move on to the humoral immunity. Now remember, there are two types of immune responses. One is called humoral immunity, another one is called cell-mediated immunity. Now what is the basic difference? Humoral immunity involves the production of antibodies, whereas cell immunity involves the cells directly going and attacking the pathogens. So let us see how the humoral immunity works. So the explanation is here. It is mediated by molecules in the blood and mucosal secretions called antibodies. Now antibodies are the proteins which are produced by specialized cells called plasma cells Plasma cells are the modified B cells, and these antibodies are responsible for destroying the pathogens. And antibodies are capable of recognizing the pathogens through their antigens. And this is one of the very effective method of destroying the pathogens and providing a specific immunity. Now this is the specialty of humoral immunity. Humoral immunity is very specific. So the antibodies produced are specific for the pathogen which has entered into the body. Now how it works? So stepwise, let us say a macrophage engulfs the pathogen. So what are macrophages? Macrophages are the phagocytosizing cells, the cells which can phagocytosize the pathogens. So it has engulfed the pathogen, and these macrophages then digest the pathogen, either whether it is a bacteria or any other pathogen, and it presents the pathogen's antigens. Now this is the most important step. You can also consider them as antigen presenting cells, because what they do is, once the pathogen enters into the system, it is going to be consumed by the macrophage cell. Now every pathogen has some molecules on its surface which are considered as antigens. Now the macrophage, once it destroys the pathogen, it is going to present the antigens. So the pathogen is destroyed, it is digested by the macrophage, but the antigens, the surface molecules of the pathogen, are presented. Now to whom it is going to present? So it is going to present these antigens to a helper T cell. Now the helper T cell is going to bind to the macrophage, and it is going to get activated by the binding. Now this T helper cell plays a role in identifying this specific antigen, and this activated T helper cell, now how it gets activated, how it gets activated, there are some chemicals like lymphokines which are involved in this process. Now the activated T helper cell is going to bind to a B cell. Now remember, there are different type of B cells which are circulating in the lymph and blood, and the T helper cell is going to bind to a specific type of B cell. And once the binding between the T helper cell and the specific B cell occurs, it will lead to the activation of the B cell. Now when the B cells are activated, the B cells are going to be converted into plasma cells. Actually, two kinds of cells are produced here. So one line, few of the cells are going to remain as memory cells, and most of the cells are going to become plasma cells. Now these plasma cells, because they have identified the antigen which is presented by the T helper cell, they are going to produce specific antibodies against this particular antigen, and these specific antibodies produced by the B cells, I mean the plasma cells, are going to be secreted by the plasma cells. So large number of antibodies are going to be secreted by the plasma cells, and these antibodies, because they are very specific for the antigen of the pathogen. Now what will happen? These antibodies will go and bind to the surface of the pathogen because of this specificity. Now binding of antibodies helps other types of blood cells to attack the pathogens. Now antibodies play a very important role by marking the pathogen cells, and they are very specific. So this is how the humoral mediated immunity works. Now, humor refers to the fluid, body fluid. Okay, so therefore, it's called humoral mediated immunity because these antibodies will be released into the body fluid. I mean, the lymph or blood. So, regarding this, again, we will watch a small video and we'll come back.
The activation phase begins when an invading bacteria is phagocytized, engulfed, by an antigen-presenting cell, APC. A lysosome containing digestive enzymes combines with the phagosome to process the antigens. The processed antigens combine with the MHC class II proteins and are presented on the surface of the APC. Helper T cells, CD4+, recognize the displayed antigen on the APC and bind to the MHC class II antigenic peptide complex. This binding triggers the APC to release the cytokine IL-1, which activates the helper T cell. The activated helper T cell releases the cytokine IL-2, which stimulates the T cell to proliferate. Thus, producing many helper T cells, each with a receptor specific for the original processed antigen. The effector phase begins when a B cell that exhibits on its surface an IgM receptor specific for the same antigen originally engulfed by the APC encounters and binds the antigen. The B cell engulfs the complex by receptor-mediated endocytosclose. The phagosome containing the antigen fuses with a lysosome. The antigen is processed. The processed antigen binds MHC class II protein and is displayed on the surface of the B cell. Helper T cells now bind to the displayed antigen on the surface of the B cell, causing the helper T cell to release cytokines. The cytokines stimulate the B cell to divide and proliferate into identical B cell copies. The B cells differentiate, change and develop, into antibody-producing plasma cells and memory cells. The plasma cells release antibodies with a specificity identical to that of the surface receptors on the parent B cell. These carry out the ultimate goal of fighting the foreign invaders. The released antibodies bind in a lock and key fashion to the antigen on the surface of the original bacterial invaders. This makes it easier for killer cells to attack and destroy the bacteria by phagocytosis, and the release of proteins causes the direct lysis of the bacteria. Simultaneously, a blood component known as complement is signaled to attack and puncture holes in the bacteria.
Okay, so the same concept is continued here. Now, During humoral mediated immunity, what happens? So the antigen presenting cell, which is nothing but a macrophage, it will engulf the pathogen which has entered into the body through any medium, and that macrophage, once it engulfs the pathogen and digests it, it is going to present the antigen. So I already told you what is antigen. So antigen is the specific molecule which is present on the surface of the pathogen. Now pathogen is destroyed, but the antigen is retained by the macrophage, and that antigen it is going to present it to a cell called T helper cell. Now helper T cell is going to receive this specific antigen from the antigen presenting cell. This is the second step. And now what happens? Once it gets the antigen, it gets activated. In the next step, what happens? The activated T cell, the helper T cell, is going to find a specific B cell. Now B cells are already there. They are either circulating in the blood or they are waiting in the secondary lymphoid organs. Now the activated T helper cell will go and present the same antigen to this specific B cell. Now remember, this activation process is actually mediated by some substances called interleukins. Okay, interleukin 4, interleukin 5, we can see here. You can think of interleukins like hormones, which are responsible for activation of the cells. Now, the B cell, which receives the antigen from the activated T helper cell, it is going to assimilate this antigen, or to be very simple, it is going to study the antigen, and then it starts producing the specific antibody against this antigen. Now this activated B cell will mature into plasma cell. Now plasma cell is the one which is secreting the antibodies. Now B cell will be converted into plasma cell, and here in this picture you can see different types of plasma cells. Now they are secreting different type of immunoglobulins. Now what are immunoglobulins? Immunoglobulins are nothing but antibodies. And there are different classes of immunoglobulins like immunoglobulin M, immunoglobulin E, immunoglobulin G, immunoglobulin A, etc. Whereas this immunoglobulin G, IgG, is the most prevalent and most common type of immunoglobulin. This is produced in maximum quantity. Now, these antibodies are very specific for this specific antigen. So they are secreted by the plasma cells. And I already mentioned some of the B cells will be converted into memory cells. Now the memory cells are the cells which are not going to produce antibodies right now, but they will remember the information about this particular antigen. Okay. Now there are two things which we have to discuss here. So one thing is about this the T helper cell, and another one is about the memory cells. Now let us see what is this T helper cell. Observe one thing here. B cell is the one which is going to get activated and which is going to produce and secrete the specific antibodies. And it is going to do that only after receiving the antigen. Now, who has the antigens? For the B cell, it is the T helper cell which is providing the antigen. But actually this T helper cell is getting the antigen from the antigen presenting macrophage. You observe one thing here. B cell is not receiving antigen directly from the macrophage. Actually, the macrophage has engulfed, digested the pathogen, and now it has the antigen, but it is not going to present it directly to the B cell. So the B cell has to get it through the mediator called T helper cell. So T helper cell is very, very important. So the transfer of antigen from the antigen presenting cell to the B cell must occur through the mediation of T helper cell only. Now, there is one condition or syndrome called acquired immunodeficiency syndrome, AIDS, which is caused by human immunodeficiency virus, HIV. Now, why HIV is so dangerous? Why it is deadly? You know what happens in the AIDS? The person is going to suffer from the secondary infections. So the person's immune system is destroyed. Now, what exactly happens there? The HIV, see, viruses, they target different types of tissues in our body. Like some viruses may target liver cells, some may target muscle cells, some may target neural cells. But the HIV virus is not going to affect any other tissue. It is directly going to destroy these activated, the T helper cells. So HIV is very dangerous because it simply destroys helper T cells. Now, once all the helper T cells are destroyed, there is nobody to transfer the information from macrophages to B cell. Since it is not getting any information, it will not produce antibodies. Even though large number of viruses are there in the body, it is actually destroying the viruses. It is trying to present antigen, but there are no T helper cells to receive this antigen, and there are no T helper cells to provide this antigen to the B cell. So the B cell thinks that there is no infection and the virus will go on destroying. Now the problem is when the secondary infection happens, like another bacteria enters into the body. So this B cell will not come to know about the infection, and the bacteria will multiply in the body and it will do its damaging things. So that is how each cell plays a very important role. That is the importance of T helper cell. Now I am talking about memory cells. So another type of cell produced by B cell is a memory cell. Now memory cells will remain in the blood for a long time. They will remain in the circulation, blood circulation, and some cells may remain for few hours, few days, few weeks, and there are some memory cells which can remain in the body for many years. Now, what is the advantage of memory cells? Now, memory cells will remember the information about this particular antigen. So, whenever the same pathogen infects the person for the second time after a gap of some months or some years, the immune response will be very quick because there is no need for studying the antigen because the information is already there in the memory cells. So that's why the secondary immune response is very quick, and the secondary immune response is very powerful. So that, okay. Here briefly we can see the difference between primary immune response and the secondary immune response. Now primary immune response is the response of the immune system when the infection happens for the first time in the life, and it includes lag phase, log phase, plateau phase, and decline phase. So here in the graph you can see this is the point of infection, and it takes some time for the immune system to respond to the infection because the macrophages are going to engulf it, then they are going to digest it, the pathogens, and then they are going to present the antigen to the T helper cells. The T helper cells in turn get activated. They are going to present the specific antigen to the B cells. The B cells will accept that antigen, they study it, and then they start producing specific antibodies. Now it takes some time, and during this time the person may suffer from some symptoms. He may show some symptoms, and after some time the person starts showing immune response. So the immune system starts showing immune response, and this immune response is in the form of increased number of antibodies, and it reaches one optimum level where all the pathogens are destroyed. And once all the pathogens are destroyed, you can say the person is cured, and now there is no requirement of more antibodies. So therefore the number of antibodies is reduced in the body fluids. Now you can see here it will never become zero, because some of the antibodies are going to remain in the blood for a long time. And there is the role of memory cells here. Suppose the person gets infected after some time. Let us say the person is cured for now, and say after 1 year, 2 year or 3 year, the same pathogen infects the person. Now this is the second time the person is getting infected by the same pathogen, and what he shows, the immune system shows, is called secondary immune response. Now you can see here the secondary immune response is very quick. There is nothing like lag phase, right? And immediately there is increase in the number of antibodies, and you can see here the number of antibodies is more than what we got in the primary immune response. So there is a quick response and there is a more production of antibodies. So therefore the person will be cured in a very short period. Sometimes the secondary immune response is so quick and good, the person may not know that he is infected. So without showing any symptoms, he may be cured. Without his knowledge, he is infected and he is cured. So that is the advantage of getting infected for the first time and getting cured. Now this graph is the one which helps us to understand the logic behind the vaccination. Now some deadly viruses, say for example polio virus or tuberculosis bacteria, they are so deadly. If the person is infected for the first time, of course the immune system will try to show the immune response, but sometimes the immune response may be delayed and the person may suffer a big loss or it may be fatal. So what they do, they give vaccination. Now in the vaccination, what happens, there is a false primary immune response. Even though he is not infected by the true pathogen, there is immune response, and the body remembers the antigen. Okay, vaccines are nothing but antigens minus pathogens. Now if the person is infected by the real pathogen, it will give him protection because now he is showing the secondary immune response. So this is the logic behind the vaccination process. Okay, so the same thing is depicted in the graphical manner here. So how the antigen is recognized and how the antigen is presented, and the clonal selection. I told you clonal selection means different types of B cells are actually present in the blood, and the specific B cells are going to be selected by the T helper cells, and those specific cells are going to multiply. That is called clonal selection, and these specific B cells will be converted into plasma cells. That is called differentiation, and now these plasma cells will start secreting antigens. Antibodies against a specific antigens, and these antibodies will do their job in the next step by protecting against the pathogen. So they are mainly involved in destruction of the pathogen. Okay. So what is the difference between cell-mediated immunity and humoral mediated immunity? Let us see some of the differences here. Now, humoral immunity refers to a component of the adaptive immunity where B cells secrete antibodies which circulate in blood as a soluble protein. So this is the main thing in humoral mediated immunity. So there is production of antibodies by the B cells. Now, whereas cell mediated immunity refers to the other component of adaptive immunity, which is mediated by the activated antigen specific T cells. So here the activated T cells are involved in the immunity. So it is humoral mediated is humoral uh, immunity is mediated by B cells, whereas cell mediated immunity is represented by T cells. Okay, so another difference is mediated by T cells, B cells, and macrophages. This is about humoral mediated, and in case of cell mediated immunity, you find the involvement of helper T cells, cytotoxic T cells, natural killer cells, and macrophages. And this is the most important difference. This is a major difference. Humoral mediated immunity acts on microbes and their toxins directly, whereas the T cell, the cell mediated immunity, attacks intracellular microbes, viruses, bacteria, parasites, etc., which have entered into the tissue cells. So, this is a major difference between humoral mediated and cell mediated immunity. Okay, other differences include the receptors. Here you find BCR receptors, and here you find TCR, T cell receptors, and this one involves the B cell receptors. And also the other receptor molecules, you find some differences between humoral and cell mediated immunity. And in case of humoral mediated, it recognizes the unprocessed antigens, whereas in T cell, the antigen is processed and then it is presented by the major histocompatibility complex molecules. And okay, this one we already understood. Antibodies are here, and here cytokines are secreted by the T cells. This is very rapid, whereas the cell-mediated immunity takes some time. So a delayed type of hypersensitivity is observed here. Okay, these are some of the important differences between cell-mediated and humoral-mediated. Very briefly, let us understand the structure of immunoglobulin G. Here I will be very brief. Immunoglobulin G is one type of antibody produced by the plasma cells, and immunoglobulin G is the one which is produced in more number and maximum quantity. So here you can see immunoglobulin G is a Y-shaped molecule. It is a large sized molecule made up of different regions. So you can see there are 4 chains actually. So this is 1, 2, 3 and 4. Now these 2 chains are called heavy chains and these 2 chains are called light chains because of their size. Okay, so this one and this one, they are heavy chains. This one and this one are light chains. Now the heavy chain is actually made up of 2 parts. Okay, here you can see the regions. This is C region, this is also C region, and this portion is called V region. Now what is C? C refers to the constant region, whereas V refers to the variable region. I told you there are so many pathogens, thousands of different types of pathogens, having thousands of different types of antigen molecules on them. So when there is an immune response, the body is going to produce specific antibodies. One antibody produced against one particular pathogen will not be effective against another pathogen. So that is specificity. Now here the specificity lies mainly in this variable region. That means you may find large number of antibodies in our blood or in the lymph. The constant region of all antibodies will be same, whereas they show difference in this variable region. That's why it's called variable region. So this is constant and this is variable. So the constant region of heavy chain is called CH, constant heavy, and the constant region of light chain is called constant light, whereas the variable region of the heavy chain is called variable heavy, and the variable region of the light chain is called variable light. Now this is the part of the antibody which is going to bind with the antigen. So that's why it's called antigen binding site. Okay. Now, okay, antigen binding and constant regions it represents. Here, fragment antigen binding, FAB, fragment crystallizable, and CL refers to the constant domain. Okay, we already studied that. So this Y-shaped molecule is commonly called immunoglobulin or antibodies, which play a very important role in the immune response. Now, earlier I already mentioned there are different types of antibodies like IgM, IgG, IgA, IgE, IgD, like that. So they show some differences in their molecular weight, and they are present in different percentages in our body. As I already told you, so 80% of immunoglobulins, 80% of antibodies belong to immunoglobulin G category, and others are also found like IgD, delta, IgE, epsilon, IgA, alpha, IgM, mu. So the gamma type of antibody is the most commonly found antibody. Okay, so I hope we understood how the immune response occurs once there is infection into our body, and for more information you can always refer other sources which are available in different places. Okay, in our next video we'll continue with the third part of this immunology topic. Thank you.